Hello students, how are you? I hope you are safe and healthy in your homes. Stay safe, stay healthy and study with me this second part of Fundamental Unit of Life. I am Mrs. Indira Chaudhary, PGT Biology from Kendriya Vidyalaya Vehicle Factory, Jabalpur. Do you remember the previous class of this chapter? We studied about the cell, if you remember. We prepared the slide of the cell in the O lab and we saw about the nucleus, etc. Let us recap. Recognize this. Which slide is this? This is the onion peel. Very good. And which cells are these? They are plant cells. Very true. And what about this? This is a cheek cell, and cheek cells are animal cells. Now these cells, these are the slides prepared in the lab. If you see cheek cell, the stain used here is blue in color. What is the stain? It is methylene blue. And here the slide has taken a red color, so that is saffronin. Now in the previous class we have studied about this darkly stained region of the cell. What is that? That is the nucleus. We have studied about the boundary of the cell. That is the plasma membrane. And here the boundary of the cell, it has the cell wall, and inside the cell wall there is a plasma membrane. So this we have studied in the previous class. We have seen the details of osmosis, diffusion, and we have seen the role of nucleus. Today we are going to study again the components of the cell, which are the cell organelles. Where are the cell organelles present? They are in the cytoplasm. If you look at the temporary stain mount of onion peel as well as cheek cell, we can see large region of each cell enclosed by cell membrane. This we have seen, and this region takes up very little stain. What is this region called? It is called the cytoplasm. So this lightly stained region is the cytoplasm. The cytoplasm is the fluid content. What is a fluid? It is neither a liquid nor it is a solid. It is the state between liquid and solid. So what is the cytoplasm is having the fluid state. The state of cytoplasm is fluid. It contains many specialized cell organelles. So let us see which cell is this. Yes, this is not onion peel. This is not cheek cell, this is a bacteria. And what type of cell is this bacteria? This is a prokaryote. If you remember that these prokaryotic cells, they do not have membrane bound cell organelles. You do not find membrane bound cell organelles here, only the DNA is here. And can you recognize what is this? Yes, some of you know what is it. It is a yes, it is the deadly virus. And which virus this is? Of course, SARS-CoV. This virus does not contain any plasma membrane, so it has only a covering of protein, but the structure like plasma membrane is not present in the virus. So they need to enter the plasma membrane of a host cell, then only they can function. So the membrane has very important role to play, and the membrane bound organelles are even more important. What type of cell is this? Of course, this is a plant cell. It will have cell organelles, and each cell organelle performs a specific function. What are the cell organelles? Let us see. Endoplasmic reticulum, the Golgi apparatus, lysosomes, mitochondria, plastids and the vacuole. Which one is the endoplasmic reticulum? This part is the endoplasmic reticulum. Which is the Golgi apparatus? This purple colored part is the Golgi apparatus. Lysosomes are small membrane bound bodies, and mitochondria, yes, correctly seen, this is the mitochondria. And what about plastids? Plastids, they are responsible for photosynthesis, so these are the plastids. And some more organelles are present. One of them is vacuoles. Which is the vacuole? This part is the vacuole. We'll study about these cell organelles one by one. So the first one is endoplasmic reticulum. What does this word endo mean? Endo means inside, and plasmic, of course, it refers to cytoplasm. And where is the cytoplasm present? It is present inside the cell. What does reticulum mean? This is a Latin word and it means network. So endoplasmic reticulum is a large network of membrane bound tubes and sheets. It looks like long tubule or round or oblong bags which are called vesicles. Let us have a look at the endoplasmic reticulum. How does it look like? This is an electron micrograph. This you cannot see with the microscope of your labs. We need an electron microscope to see this. So the scientists have given this structure. And what is this? Is it endoplasmic reticulum? No, this is the nucleus. If you remember about the nucleus has two membranes. These are the two membranes of the nucleus, and these membranes are interrupted by the nuclear pores. So many nuclear pores are present, you can see here. One of the pores is entering this tube-like structure, tube and sheet-like structure. This structure is endoplasmic reticulum. This is looking a bit different, and have a keen look at this. Yes, this is the endoplasmic reticulum. Reticulum means it is forming a network inside the cell, and it is occupying a lot of space in the cell, and it may occupy this space right from the nucleus towards the plasma membrane of the cell. Let us see how many types of endoplasmic reticulum are there. Yes, there are two types of endoplasmic reticulum. One is rough endoplasmic reticulum and other is smooth endoplasmic reticulum. We can draw a diagram like this, that this is the endoplasmic reticulum, and this has small structures attached here. What are these small structures? They are ribosomes. So RER looks rough under the microscope because it has particles called ribosomes. So these are the ribosomes attached to the endoplasmic reticulum. What do these ribosomes do? These ribosomes, if you see again the electron micrograph, they show a structure like this. So these are the ribosomes, and they have a very important role to play. They are acting as the ribosomes, which are present in all active cells, are the site of protein manufacture. This is giving a clue that proteins are manufactured here, and this ribosome proves to be the site of protein manufacturing. These manufactured proteins are then sent to various places in the cell depending on the need. The cell needs lot of proteins, fats, lipids, and these are used at various places. So the manufactured proteins are used by the cell. Let us see what does the smooth endoplasmic reticulum look like. This is the smooth endoplasmic reticulum. You can draw a diagram like this, and these smooth endoplasmic reticulum, see, they do not have any ribosomes. They manufacture fat molecules. These smooth endoplasmic reticulum they manufacture fat molecules, or we call them lipids. They are important for the cell function. Do you remember? Where do you see lipids? These proteins and lipids help in building the cell membrane. In the previous class, we have studied the structure of cell membrane, which is made up of proteins and lipids. So the cell is very independent. It can produce its own proteins and lipids, and then it can build the cell membrane. And when the cell membrane is built by the cell, this process is known as membrane biogenesis. If the cell membrane is damaged, the cell can repair its membrane by process called membrane biogenesis. What is the role of these proteins and lipids? The proteins usually function as enzymes. We'll see what are enzymes in our classes. And proteins and hormones also form, proteins and lipids also form hormones. Now, endoplasmic reticulum varies greatly in appearance in our different cells, but it forms a network in all the cells. So, what are the functions? Let us recap the functions of endoplasmic reticulum. Yes, it serves as the channel for transport of material. Since the materials are synthesized, the proteins are synthesized, now they need to be transported for the use. So, they are transported between various regions of cytoplasm or between the cytoplasm and the nucleus. This endoplasmic reticulum functions as cytoplasmic framework, providing a surface for biochemical activities. Now, whatever is formed, proteins or lipids, they perform some biochemical activities. So, this endoplasmic reticulum is the surface for that. In liver cells of vertebrates, do you know what are vertebrates? These vertebrates are animals having backbones. These are animals with a backbone. So, these animals, the endoplasmic reticulum which is present in the liver, they have a very important or very crucial role to play. What is that? To detoxify many poisons and drugs. In animal body, many toxins, many poisonous substances or drugs may enter. So, they can be detoxified. It means those poisons and drugs can be neutralized. That process is called detoxification. This is done by what? In smooth endoplasmic reticulum. So, this is very important. It keeps the organism safe. And of course, the last function is formation of cell membrane. What is that process called? It is called membrane biogenesis. So, let us look at this. This is the endoplasmic reticulum. You can see here, this is the rough one and this is the smooth one. The next cell organelle is the Golgi apparatus. This is a peculiar name. Okay, can you tell me why it is called Golgi apparatus? It was named after a doctor, Camillo Golgi. He was an Italian doctor who studied in Pavia, and he was studying the neuron cells. And while studying the cells of the brain, he developed a technique of black staining. And there he saw first these structures called Golgi apparatus. So, in the honor of Dr. Camillo Golgi, we call this organelle as Golgi apparatus. As you can see, it looks like a system of membrane bound vesicles which are flattened sacs. These are flattened sacs and they are arranged approximately parallel to each other in a stack called cisternae. These are cisternae. They have connections with the endoplasmic reticulum, and let us see how are they connected. If this is the endoplasmic reticulum, then this is connected to this Golgi apparatus. What is the role of Golgi apparatus? whatever material is synthesized in the Golgi apparatus, it is packed and dispatched to various targets inside the cell and outside the cell through Golgi apparatus. So what is the function? One of the major functions is storage, then modification. It means it can change the proteins or the lipids, then packaging of the product in small bags called vesicles. These are small vesicles. In some cases, the complex sugars may be made from simple sugars in the Golgi apparatus, and it is having a very important role, that is formation of lysosomes. We'll study about the cell organelles, lysosomes, but let us have a look how does Golgi apparatus perform its function. This is the endoplasmic reticulum and this is the Golgi apparatus. How do they function? Let us see. Some storage, then modification, and this is how the vesicle is formed. Some storage, modification and packaging. This is how small vesicles are formed by Golgi apparatus. You can have a watch, storage, modification and then packing. You can have a look here. You should try to draw these diagrams. What is this? This is the nucleus, and from here the endoplasmic reticulum arises. It is a dense network covering a large area of the cell. This is what you have studied till now. So you can practice making these diagrams and just attach these small ribosomes here, and this is the Golgi apparatus. Try this after this chapter. The next organelle, here also you can see how these vesicles are formed, and this Golgi apparatus packages them and then it is used in various places. So they are working in collaboration to each other. It is a teamwork. It is something like you have a team in sports. Everywhere it is a teamwork. This is the nucleus, then this is the endoplasmic reticulum. It is connected to the Golgi apparatus. Golgi apparatus is helping the cell. So this is how they all function together, and cell becomes the structural and functional unit of life. The next cell organelles are lysosomes. Where are they formed, lysosomes? Yes, they are membrane bound bags filled with hydrolytic enzymes, and they are formed in the Golgi apparatus, as we have seen. Let us see what is the role of these lysosomes. They are filled with hydrolytic enzymes. What are enzymes? Enzymes are, we can call them digestive enzymes, and as the name signifies, they perform a function of digestion. What is digestion? Digestion is breaking of larger molecules to smaller molecules. So these enzymes are made by endoplasmic reticulum, and these lysosomes store them. And what do? They have a kind of waste disposal system. How? It keeps the cell clean by digesting the foreign material or the worn out cell organelles. Whatever enters the cell, if that is a bacteria or a food material or a worn out cell organelle, then these lysosomes will combine with that and break these complex substances into simple ones. How? Because they have these powerful digesting enzymes. These are digestive enzymes. Now, suppose this is lysosome inside the cell and there is a debris. So, this is a debris. It is a broken part, or say it is a bacteria. So this lysosome is going to merge with this and it is going to form a big vacuole like this, and it is going to digest it. So this is the process of cleansing. Then if a lysosome during the disturbance in cellular metabolism, if the cell gets damaged, then the lysosomes can burst. And since they have the enzymes, digestive enzyme, can it digest its own cell? Yes, they can digest their own cells. So if this lysosome breaks inside the cell, this lysosome is inside the cell and it can digest its own cell. So what will happen to the cell? Of course, the cell is going to die. That is why we can call these lysosomes as suicidal bags. This is an accidental condition for a cell, that it can perform suicide of a cell. Let us see how this functions. Again, this part is the nucleus, this is the endoplasmic reticulum, this is the Golgi apparatus. Now this Golgi apparatus has made many lysosomes, and this is a debris. Let us see what happens. See, the lysosome attaches and it is digested. The lysosome attaches and the digestion of this debris takes place. This is how the lysosomes function as the cleansers. The next cell organelle is the mitochondria. It is looking very powerful. Yes, this is mitochondria. And commonly, what do we call these mitochondria? We call them powerhouse of a cell. What is a powerhouse? At your homes, the powerhouses give you electricity, the power to do work. Your fans, your ACs, they work by power, electricity. So this also gives the power to the cell. Let us see the structure of this mitochondria. What do you see here? What is this part? Of course, this is the membrane. Which membrane? This is similar to the plasma membrane. We are studying these membrane bound organelles. So this is the outer membrane. This is the outer membrane. You can see that it is regular and it is porous. But what about the inner membrane? This inner membrane has infoldings. You can see here the inner membrane has foldings. So these foldings, they are called cristae, and inside this folding, what is present is called the matrix, which contains numerous enzymes for respiration. So this is the mitochondria. Let us look into the detail of the structure of mitochondria. As I told you, they are called the powerhouses of the cell. And how many membranes are there? Mitochondria have two membrane coverings, the outer membrane and the inner membrane. The inner membrane is deeply folded. These fold, what do? They increase the surface area for what? For ATP generating chemical reaction. So what are these ATPs? These are energy rich molecules. The energy required for various chemical activities needed for life is released by mitochondria in the form of ATP. So this ATP are energy molecules, and these energy molecules, we call them currency of the cell. The ATP is known as currency of the cell. What is a currency? What is the currency of India? Rupee is the currency of India. So if you want to buy a book for yourself, you need money. Similarly, if the cell has to do work, the cell body uses stored energy stored in ATP for making new chemical compounds. So every chemical reaction requires ATP for making new chemical compounds and also for mechanical work. What is a mechanical work? For example, you work with your hand, You write, you work, you walk. This is done by the muscles, and the muscles get energy in form of ATP, which is generated in the powerhouse of the cell. These mitochondria, they are a bit strange. Why? Because they have their own DNA. The cell has a nucleus, the nucleus has the DNA, and what is the role of DNA? DNA is the genetic material. DNA is genetic material. So, these mitochondria, they also have DNA inside them. So, they can also perform some functions, and they have ribosomes. So, they can make their own proteins. So, it is sort of independent and autonomous. So, it can make their own proteins. You can see this is a mitochondria. This is the outer membrane, which we have already seen. And what about the inner membrane? This is the inner membrane. This has foldings. So, this is the inner membrane. And this inner membrane has many functions, many chemical reactions, and they generate the ATP. Let us see how the cell does it. The ATP or the power. See, the cell can generate power. And what are the power molecules? They are the ATP. So, the mitochondria is giving ATP to the cell to do work. Similar next organelle we are going to study is the plastids. These plastids make the world colorful. These plastids are present only in plant cells. The plant cells have plastids, and these plastids are of two types. One is a chromoplast and one is the leucoplast. Why do we call it chromo? Chroma word here means color. So they are colored plastids. Plast word is referring to the plastid, and chroma word is referring for color. So the colored plastids, and the leuco word refers to white or colorless. Plast again is related to plastids. So leucoplasts are colorless plastids and chromoplasts are colored plastids. Whatever colors you see in the nature, they are because of these chromoplasts. And this earth is called the green planet. The greenery we have on this planet is because of the green plastids. So chromoplasts, which contain pigment chlorophyll, are known as chloroplasts. So chloroplast is actually a type of colored plastid which has green pigment called chlorophyll. And these chloroplasts, they have a very important role to play, that is photosynthesis. What is photosynthesis? Can you tell me? Yes, photo, as the name signifies, photo means light. And what does synthesis mean? Synthesis is to produce. So prepare. So by use of light, they can prepare what? Food. By use of carbon dioxide, water in presence of sunlight. Let us see how do the cells do that. The cells are making use of light and producing the sugar molecules. Let us see the structure again here. This is the chloroplast, and this part is the outer membrane. One more membrane is present here. Yes, this also has two membranes, but this membrane is not folded like this mitochondria. They are straight, and this is the outer membrane and this is the inner membrane. So this chloroplast also has 2 membranes. This is the beautiful colorful world of the flowers, and the flowers are colored due to which plastids? Chromoplast, correct? And the leaves are green due to the green plastids, the chloroplast, and the flower, the red colored flower, Delaunix regia, you can see here, and this is having chromoplasts. Leucoplast, they store starch, and they are storing oil, starch, proteins in potato, in vegetables. Chromoplast, they store color pigments, either you see the outer covering of the brinjal, or you see the carrot or the flowers. The chloroplast, the basic function of chloroplast is to perform photosynthesis, and leucoplast mainly store the food material, and chromoplast are for imparting colors. The next cell organelle we are going to study is vacuoles. You can see these are the vacuoles. Which cell is this? This is a plant cell, correct? And which cell is this? This one is an animal cell. And let us see the position of vacuole in plant cell. This is looking quite big, and it is almost parallel to the cell wall, so it has some special functions because of this structure. So the plant cell contains large central vacuole which is filled with water, and it helps to give shape to the cell. So the stable shape of a plant cell is because of this big water filled vacuole. Let us see the vacuoles of this animal cell. They are quite small, and they store water and food materials. Sometimes they store the waste material also, and help the cell to get rid of these waste materials. So in short, we can say that these vacuoles are sort of warehouses, store warehouses which store water, food material, and sometimes the waste material, so that the plant can store them or it can get rid of them. So these were the cell organelles which perform very important function in the life of a cell, and cell is the part of any multicellular cell division. This is the new topic for this book. NCERT book has introduced cell division for the first time. It was not here in the book previously. Now what is this cell division? It means a cell can divide. Why does a cell divide? Why does it need to divide? New cells are formed in organisms in order to grow and replace old, dead, injured cells, and also to form gametes required for reproduction. Now here it is organism. So organism is multicellular, and this multicellular organism has the unit of cells. So these cells of this multicellular organism can divide. Why? To grow, to replace old dead cells, and also to form gametes required for reproduction. So this is the process of cell division. This cell division is of two types, mitosis and meiosis. So we can say that a cell divides by two methods, mitosis and meiosis. Let us see what is this mitosis. In this process, most of the cells divide for growth, and that is by mitosis. How? There is a mother cell, and that mother cell divides into daughter cell. We don't call father cell and son cells, but since they give rise to the new cell, so this is a mother cell, and this is mother cell will divide into two daughter cells. How many? This is very important, that in mitosis a mother cell divides into two daughter cells. One more condition is there, that the daughter cells have the same chromosome number as that of a mother cell. And if, let us see, if this is a cell and this cell is dividing into two daughter cells, so if this is a single cell and this single cell has the nucleus, and the chromosome number of this nucleus, say if it is a human cell, it is 46. So this cell has 46 chromosomes. So which cell is this? This is the mother cell. and this mother cell is going to divide by this process. So this one, these are daughter cells. So this is going to give rise to daughter cells. This is one daughter cell and this is another daughter cell, and these daughter cells should have 46 chromosomes. So this daughter cell and this daughter cell, the chromosome number should be 46 here, it should be 46 here, and the mother cell should also have 46 chromosomes. Why this mitosis? We have seen already, it is for growth. You get injured, sometimes you get a cut, and when you have a cut, it is like this, and when it is repaired, again it is smooth. Why? Because of mitosis. So these are the cells of your skin which divide by mitosis and repair your wounds. You remember when you were a small child, you were not in the school, so you were of this size. Then you grew, now you came to class 1, now you are in class 10. So what is happening? The child is growing. What is this growth due to? This growth is due to division mitosis. Let us look at a plant. This plant was very small and now it is going on increasing in height. The size and shape of the leaves is increasing. Why? Because of the cell division mitosis. So the importance of mitosis is growth and repair. This is how a plant grows. A plant cell by cell divide and finally it forms a very big tree. This is all by mitosis. Next division is meiosis. The specific cells of reproductive organs or tissues in animals and plants divide to form gametes, which after fertilization give rise to offsprings. They divide by different process called meiosis and which involves 2 consecutive divisions. Now this takes place in reproductive organs and form gametes, which after fertilization give rise to offsprings. And there are 2 consecutive divisions in this meiosis, and 4 new daughter cells are formed, not 2. In mitosis, only 2 daughter cells are formed, but here 4 new daughter cells are formed from one mother cell. The new cells have half the number of chromosomes than that of the mother cell. If this is a cell, human cell having 46 chromosomes, so it will divide by 2 consecutive divisions. One is this and another is this. Another division will give rise to again 2 each, so 4 daughter cells. The chromosome number in the daughter cells has to be half, that is 23, 23, 23 and 23. So this is how the chromosome number is reduced half. Why do you think that this chromosome number is reduced half? This is taking place in, say, plants or animals. If it is plants, it takes place in the stamen and the pistil. If it is in animals, it is taking place in either the testis or the ovary. In each case, the mother cell, if it has 2n number of chromosomes, then the number of chromosome is reduced to half in the daughter cells. Either it is an animal cell or that's a plant cell. So the chromosome number has to be reduced to half in the daughter cells. What is the reason behind this? That the mother cell has double number of chromosomes and the daughter cell have half the number of chromosomes. There is a specific reason behind that. In case of sexual reproduction, there are 2 parents. One is the mother parent and other is the father parent. Now mother parent will give rise to a gamete and father parent forms gametes in the reproductive organs. The gametes formed will have, the cells of the reproductive organs will have 46 chromosomes, but the gametes formed will have 23 number of chromosomes. Now ovum has 23 cells and sperm has 23 cells. Now they unite and fertilize and form the zygote, which has 23 plus 23 total 46 chromosomes, which is the number of the cells found in mother and father. So this zygote has the chromosome number which is found in all the cells of the father and all the cells of the mother. But the ovum and sperm are special cells. They maintain the chromosome number. Why? Because each species has a constant chromosome number, and species maintain constant chromosome number by process of meiosis. Let us perform a practical which we started initially, and that is to prepare a temporary stain mount of human cheek cell.
To prepare a stained temporary mount of human cheek cells, to do the experiment we require toothpick, cover slip, methylene blue solution, glycerine, glass slide, distilled water, brush, needle, compound microscope, filter paper, and a dropper. Procedure: pour some distilled water from a bottle in the middle of a dry glass slide. Take a clean toothpick and gently scrape the inner side of the cheek. Rub the cheek tissue on a glass slide that contains water. Mix the mixture using a needle and spread it. Take methylene blue solution using a dropper and add a few drops of it to the mixture on the slide. After 2 to 3 minutes, remove excess water and stain using a blotting paper. Take glycerine using a dropper and add a few drops of glycerine to the test mixture. Take a clean cover slip and lower it carefully on the mixture using a needle. Using a brush and needle, press the cover slip gently to spread the epithelial cells. Remove extra liquid around the cover slip using a blotting paper. Take the glass slide and place it under a compound microscope. View the slide under the compound microscope. Observation: a large number of flat and irregular shaped cells are observed. The cells do.
Let us recap the chapter. What we have studied till now in this chapter: we have studied the specific functions of the cell organelles, which are endoplasmic reticulum, which is responsible for protein synthesis and lipid synthesis. Golgi apparatus, it is for storage, modification and packaging. Lysosomes, they contain digestive enzymes which are responsible for the digestion inside the cell of the debris. Then the organelles, mitochondria, the powerhouses of the cell which produces ATP by respiration. Plastids, the chloroplasts which perform the function of photosynthesis. Plastids impart color. And the vacuoles, which are the storehouses of water in the cell. Then we have studied about the cell division. The cell division is of either mitosis or meiosis. Mitosis is an equational division where only 2 daughter cells are formed, with mother cell and daughter cell having same chromosome number. Meiosis, this takes place in reproductive organs where 4 daughter cells are formed, with daughter cells having half the number of chromosomes. After that, we have prepared the temporary stain mount of human cheek cell, and now what is required is home practice. What we studied in this chapter, this was the discovery of hundreds of scientists in hundreds of years. So you have the complete knowledge, explore it, enjoy it, find it out and practice it at home. Finally, stay healthy, stay safe. Thank you very much.